Okay, so here we are going to go over labs 4.1 and 4.3. You'll need to know this information because you'll have a quiz on this exact review sheet tomorrow. Number 1. How did we genetically engineer bacteria to produce a glow-in-the-dark protein called GFP? Our answer here is the jellyfish gene was first spliced into plasmid DNA, and then we inserted the plasmid DNA into the bacteria. How is this process very similar to how human insulin is produced for diabetic patients? Your answer is the human insulin gene is spliced into plasmid DNA, and then the plasmids are inserted into the bacteria. The bacteria will then produce human insulin protein for patients. How did we insert the plasmid into the bacteria? Your answer here is we called it the heat shock method. This was used to kind of freak the bacteria out and give them that drastic temperature change. Bacteria were first placed on ice for about 10 minutes, then they were placed in the hot bath set at 42 degrees for about 50 seconds, and then you put them back on ice for the last 2 minutes. This allowed them to take in the plasmid DNA that was around them. What was done to the plasmid by genetic engineers prior to our lab? So this is the part we could not do by ourselves. So your answer here is the jellyfish gene was spliced into the plasmid after both were cut using the same restriction enzyme in order for the sticky ends to match up.
Here's a short video of me explaining again what took place in the
Here's where I'll explain the recombinant DNA that took place during our pGlo lab. Since all DNA, whether in you or a blade of grass or a bacteria, are all made of A, C's, G's, and T's, theoretically any DNA can be recombined together. So in our lab, we took DNA from a bacteria, in this case it was the small plasmid ring, and then we also took DNA from a jellyfish, because they had this target gene of this green sequence that produces this green glow protein we wanted the bacteria to produce. It's the same exact procedure that's done to grow human insulin to give the life-saving medicine to diabetic patients. So we have to first cut the DNA before we can at all recombine it. So with that, we have to use these molecules called restriction enzymes. They act like molecular scissors that scan a DNA sequence and make cuts along the DNA whenever they find this particular sequence. So we have to use the correct restriction enzyme in order for the ends to match up correctly. So what we're going to do is apply one of these two. In this case, only the Hindi3 restriction enzyme will work the best. And we'll apply it to our target gene. It'll find the sequence, make the cut. We then therefore have to use the same restriction enzyme over here on our plasmid DNA to locate the sequence and make the cut. Now, since we've used the same enzyme, we can take the two molecules and splice them back together, allowing their sticky ends to bind together. Here you see C and G's are opposite, A and T's are opposite, so the DNA will be recombined. So here, this is now our recombined DNA that we then heat shocked and added back into the bacteria in order for them to produce that protein. Now, if they have that gene, RNA polymerase can come by, code for the glow protein, and produce that glow protein that actually glows. The sequence itself is not any color, it's just DNA, but the protein that produced glows. And then here you have DNA from two sources combined into one organism. Jellyfish DNA and bacteria DNA all together inside of the bacteria. That's it. Explain why the bacteria needs sugar in order to express the GFP, even though both plates had the GFP sequence inserted. So remember here, in this case, sugar has no nutritional value for bacteria in this case. They're feeding on that brown nutrient agar. The sugar, however, removes the repressor that was blocking the gene from being expressed and allowed RNA polymerase to code for the gene and make the GFP protein. So even though both plates received the gene, only one of them was able to express it.
Here's a quick little video summarizing what took place and how that sugar removes the repressor.
Bacterial DNA, a repressor, mRNA, and the sugar molecule lactose. Gene expression can be induced or turned on when a specific inducer molecule appears in a cell. For inducible systems, a repressor molecule prevents gene expression by binding to the upstream controlling region. Lactose passively enters the E. coli cell and binds to the repressor molecule. This binding releases the repressor from the controlling region. At this point, RNA polymerase can begin transcription of the operon. Here we show 2 of the 3 lac operon genes being transcribed into mRNA. Ribosomes then bind to the mRNA, and the 2 proteins are translated.
So here in this slide we're showing that both plates have the gene in the bacteria, however only the sugar one is able to remove the repressor and allow polymerase to transcribe the gene. Why can't we just inject a person with the bacteria that are producing insulin? What was the point of the protein column chromatography lab? Your answer here is we needed to separate out or purify the one protein we need from all the other bacteria tissue and the other proteins they have produced. So we loaded all the cellular tissue and proteins into the column, and as they dripped through the column, we were able to sort them out by size. So here in our lab 4.3, this was the protein column chromatography lab, in which we purified out or separated out all of the cellular proteins that were produced by the bacteria. The only one we cared about was the only one we were looking for, and that was the GFP, the glow fluorescent protein. So here are 5 fractions from that lab. You collected 8. These are 5 of those. You can see here in tube number 2 that the GFP is present a little bit because the tube is kind of glowing just a little bit as it was just starting to drip into the tubes. The larger proteins come out first, so 1, 2, 3 has larger proteins, 4, 5, 6, 7, 8 would have smaller proteins. Then you see over here in tube number 4 that there's still a little GFP that was on its way out and still kind of dripping through the column as the tube is still slightly glowing. However, here in the middle of tube number 3, this has clearly been the purified tube that contains the pure GFP that we care about. So in the case of human insulin production, although insulin does not glow in the dark, once you've engineered bacteria to produce insulin, this would be the tube that we would process and give to patients. Compare our protein column chromatography lab with the process of SDS-PAGE. So 8A, what is the same? Well, both processes here sort proteins by size. The difference here in part B is that smaller proteins take longer to travel through the column in chromatography, and they come out last, compared to SDS-PAGE when they were first. So in the column chromatography lab, the larger proteins fell out first. So in your fractions 1, 2, and 3, those were the bigger proteins, and then in 6, 7, and 8, those were the smaller proteins. However, in an SDS-PAGE gel, those smaller proteins travel faster and farther down through the matrix and are located down near the bottom of the gel, and the larger proteins are stuck up towards the top. Draw a conceptual graph of the GFP concentration, supposing that the 4th fraction glowed the brightest. So here's our graph. We see that the protein is starting to come into play in fraction number 3. It's clearly mostly concentrated in number 4 because it's glowing the brightest. And it's still present in fraction 5 and maybe even a little bit in 6. So 3, 4, 5, and 6 are probably all glowing. It's just that 4 is definitely glowing the brightest as it has been purified. So since insulin doesn't glow, how do you suppose researchers check to make sure the 4th fraction really only contained the protein of interest and nothing else? How can they make sure it was truly purified? So since insulin doesn't really glow, after you were done running a chromatography, how would you know exactly which one contained it? You would therefore have to run an SDS-PAGE gel and make sure that only one band shows up in the tube you think the protein is in. This would then therefore prove that you have purified the one protein out you want from all the rest. If more than one band showed up in the gel, you would know that other proteins were mixed in there.